Good afternoon and welcome everybody. Thank you for joining us today. I hope that many of you got a chance to review the subject matter of the webinar today. It is fascinating, at one time science fiction and no longer science fiction. You're going to discover really quickly how extraordinary this is. And as many of you know, I have no skin in the game at this point, although I may. So I'm not being hyperbolic. This is really fascinating stuff. And I've had a chance to see who is logged on and registered for the webinar. And I welcome all of you because many of you I know, and also many of you are very involved in this type of science. I also want to welcome our dear friend, who many of you know and love, Wendy Kraft, who will be hosting and co-hosting many of the Family Office Insights webinars, and I welcome her and delighted that she's going to join us today as well. Thank you, Wendy, for being here.
Arthur.
So just a little bit of housekeeping. As you know, we can't see you or hear you, but we encourage and welcome questions in the Q&A in the chat box. And we'll be getting to them as many of them as possible. We have a lot of people that have signed on today, so don't be shy. Go ahead and put your questions in, and we'll, as you think of them, and we'll get to them as time permits. We have an hour today. For the record, we're going to be recording this, so if you care to review it or share it with others that might benefit from it, you can find it in a couple of days on the Family Office Insights YouTube channel. And thank you, Iva. And so it'll take a day or so to upload it, but it'll be there. And of course, as usual, we'll make sure that Rene is in direct contact with you post-webinar, and he'll have the list of all the people that registered and joined today. So again, welcome. Thank you for spending your afternoon with us here for an hour. And with that, Wendy, just say hi real quick because everybody knows you.
Good afternoon, everybody. Thank you so much for joining us. It's a pleasure to have you here.
Great, thank you. So Rene, we're going to go mute and off our video. And so if you can go ahead and get started, and welcome and thank you very much for not only sharing with me prior to this call and a couple of phone calls, the fascinating stuff you're doing, but joining and sharing it with the Family Office Insights community today. Welcome, Rene, take it away.
Thank you, Arthur. It's a privilege to be on your Family Office Insight Forum. We're grateful for giving us this opportunity. I am truly delighted to be sharing this with a larger community of investors and others that are in the audience today, and thank you for your attendance. Just a little bit about my background. I am a neuroscientist, a genomist, and also a chemical quantum trained scientist. So what you will see is in the work that we do, we bring all of these different talents together to bear on problems relating to brain disorders. I was a faculty at the University of Pennsylvania. I trained at the Salk Institute. I am currently running this business as an entrepreneur. And it's really, truly exciting to be an entrepreneur bringing bench science to the bedside. I started Neuxstem in 2015, and we have progressively built up the business. And that's what I'm going to share with you as to where we are in the process of bringing this to the clinic. So Neurxstem is a predictive and precision genomic brain medicine company. And our core goals are the diagnosis, early diagnosis, and effective treatments of human brain disorders and diseases. We are developing big data analytics for brain disorders, and I'm showing you a quotation from a recent issue of Pharma Voice, where they claim the future of big data analytics isn't just bright, it's the future of medicine. And that's what we're trying to usher in, and that's why we are so excited. To give you some sense of where the business of precision science is, I've given you a couple of benchmarks of where things stand today. There have been very large investments in precision medicine, and you can start to see that in combination, data in combination with sophisticated machine learning or AI or other algorithms will open the door to substantially de-risking and shortening the drug discovery process, and also truly understanding what underlies the pathology in many brain disorders. Our vision of where I think we should be going is derived from the work that came out of the Nobel laureate Shinya Yamanaka, who in 2012 showed that you could take skin cells and make them into stem cells, pluripotent stem cells. And that opened the door for many, many groups trying to make different organs. We are interested in the brain to tackle issues to do with brain disorders. Neuxstem has two major goals, business goals: diagnostics and therapeutics. And we have multiple verticals: cancer, brain cancer, substance use disorder, neuro stress trauma, neurodegeneration like Alzheimer's disease, and neurodevelopmental disorder. How is it we can do so many different verticals? It's because we have developed a platform technology that allows us to access all of these diseases all at the same time. Where are we located? We are located in Heath, Ohio. And if you haven't been to Heath, Ohio, it actually houses one of the largest Boeing facilities and the Air Force facilities are right next door. So we are nested amongst aeronautical engineers and all sorts of engineers, and we are the only biomedical firm in this location. Our goals are inspired from aviation. In 1903, this is what innovation looked like. And I don't have to tell you that nobody could have imagined what it would look like today. And I'm using this in the metaphorical sense because this is the giant leap we're going to take. And not on the same time scale, but on a much shorter time scale, we'll be reaching the kind of things we have seen in aviation. In 2005, we disclosed that we had made the full complete neural organoid, which is a mimic of the human brain from skin cells. And this is how the press responded to our innovation. We were on CNN. It was called a quantum leap forward for many brain disorders and cancer. BBC covered us, Radio Australia covered us, and a large number of press releases were out there. So where have we gone since 2015? We have begun to address, using the model system, the staggering brain health problem. And that has to do with, let me show you here, I'll turn on my laser pointer, the cost, incredible cost that we're bearing right now for cancer, autism, dementia, addiction, and PTSD, but more so lost wages, mortality, and suffering. Today, PTSD, unfortunately, takes the lives of 22 veterans a day. The opioid crisis has caused the deaths of over 800,000 people. We are addressing all of these almost all simultaneously. So we're very glad the technology has a bearing on multiple domains of brain sciences. What is the big problem? There are no effective therapies for any of them. Why was that the case? That is because we made a deadly assumption that rodent models were the equivalent and would serve as good proxies for human brain disorders. Unfortunately, it had disastrous consequences for the patients with brain disorder and diseases. We have seen no real solutions for any of them. And so our premise was to change this whole trajectory almost 10 years ago. And we began the process of engineering a human brain model solution for early diagnostics for the prevention and new drugs for effective therapeutics. It's a singular solution with vast reach across many disorders. So from a business standpoint, we have created what I call NNOP, which is Neurxstem Neural Organoid Platform. And that is the primary offering from a business perspective. It starts with skin cell, the proprietary product, molecular fingerprinting, the generation of proprietary big data analytics. That opens the door for early diagnostics and therapeutics. The areas that will impact goes from obstetrics, pediatrics, neurology, neuro-oncology, and psychiatry. And the product is a neural organoid that I'm going to talk about a little more right now, which is shown here on the left side. That's the neural organoid. That's a proprietary product and proprietary analytics that are coming out of it. The brain disorder, the neural organoid, when it comes from a patient, actually produces brain disorder organoids that are bioengineered in the dish. They are typically about 4 weeks, the equivalent of 4 weeks in development of the human brain, which is what you're seeing over here. And they don't quite look like the human brain. They look like something different, which I just showed you earlier. And what the evolution of brain sciences has been from postmortem to MRIs to now what we think is the future, which allows us to look at the development of the brain and disease trajectories in real time in living, growing model of the brain to look at changes in gene expression, metabolites, and proteins. Very, very powerful big data is coming out of it. And so it's the brain in a dish, and it's 98% identical to the adult human brain. And that is stupendous because it recapitulates everything that happens in a disease state. And I'm just going to show you a vignette of the gene expression and development across weeks, days, and hours. And so this is a markup of the neural organoid. And we have data from the retina, from the cortex, from the midbrain, from the cerebellum, from the spinal cord. So the entire thing from one end, eye disease, to that of the spinal cord this model can be used to study and understand any disease process involving the brain. So when we do that, this is what these organoids look like. This is a large number of them in a dish. And that is a high magnification of what they look like with all the canonical structures of the brain that I showed you earlier. And I'm going to hold up a real life demonstration of what this looks like and tell me if you can see. I'll have to cover the light a little bit.
I think just actually that a little closer.
Right
there.
Can you
Right
see that
there.
white little blob there? I'm sorry, the light's reflecting a little bit on it.
Yeah.
Give me a second. I think this is so magical to see this. You can see that now?
Yep.
That white blob? Well,
Yes.
that is a human brain from a schizophrenic patient that has been preserved, and that's about the size of the product that I'm talking about, the model system. It's got all the structures in there under magnification and when we look at it using various techniques, that's the bioengineered brain. So when we take this bioengineered brain from two different model systems, one is normal and one is diseased, we are able to then look at the changes in gene expression across all the genes in the human genome. There are about 21,000 genes. As an example, I just took 4 genes here, and 2 of them are dysregulated. It's the change in gene expression that is what we measure to understand the disease process throughout the development of the brain. And here's data where we are looking at 13,000 genes that are expressed in the human brain out of the 21,000 from replicates from Alzheimer's, normal, and a disease called tuberous sclerosis, which has both tumors and cancer and autism. And the beauty is when we look at the differences between these is when we start to pick up the biomarkers. What is important to note is for a business application, a clinical application is reliable, you need data sets where all of the data is a virtual straight line, as you can see here. There are about 12, 13,000 points here, all sitting on top of each other. And that power comes from the meticulous way in which we develop these organoids and QC them and make them in a manner such that they are useful for the kinds of end goals that we have. From that comes out a large number of diagnostic and therapeutic targets combined. And for opioid crisis, we have about 50 right now, Alzheimer's 2,000, Parkinson 70, schizophrenia, autism, and brain tumors, and also multi-organ cancers that share biomarkers with the brain tumors that we study. The predictive precision genomic brain medicine examples, I'm going to take you through just examples, Alzheimer's disease. When we look at the differences, this is what we see. We see a swath of genes that are all innate immune system genes that are dysregulated And we predicted that in 2016 using our Alzheimer's model, that the skin cells that we used came from an Alzheimer's patient. 1 year to 2 years later, these same discoveries were made by others in postmortem brains and using genetics. They also starting to see that one driver of Alzheimer's disease is the immune system. So this tells you the power of predictive medicine in a dish versus what happens in real life, which gives us the corroboration, gives us confidence we're looking at something that is real. Similarly, we have now done a full analysis of the Alzheimer's disease. This is patented information. And here are the comorbidities that are known clinically in Alzheimer's: retinal disease, heart disease, impaired immunity, psychiatric illness, on and on and on. And here are all the exact genes that are involved and how they're dysregulated, and whether they are or aren't expressed in the postmortem. 90% of them are. And here's all the references that tell you that we are making a one-to-one map within 4 to 6 weeks of what will happen to you 50 to 60 years from now. That's profound. And I picked this for a reason, because we believe that the susceptibility markers for Alzheimer's exist at birth. Why it plays out over time, I can get into the details, but it has to do with the other 2 other pillars of disease that I'll talk about shortly. Here's an example of autism spectrum disorders. We did the same thing. In 2016, we predicted there would be zinc deficiencies, magnesium deficiencies, lead toxicity. And that's why nutrition and toxins were also a contributor to the disease. In 2017 and 18, a beautiful study comes out of the Karolinska Institute in Sweden. They're looking at baby teeth from twins that are monozygotic twins, but one has autism and one doesn't. And the biggest differences in the teeth were the accumulation of lead, the lack of zinc, and lack of magnesium in their teeth, baby teeth, in the child that was diagnosed with autism. So you can see, whilst they know this, we know exactly why, based on the biomarkers, why these things are occurring. So it gives us unprecedented views into what we need to tackle and why we need to tackle it. And similar stories for cancer. This is the MRIs of tumors in the brain, and be able to pick up biomarkers, well-known biomarkers for brain cancer that are dysregulated to very large degrees in our model system. So with these 2 examples, what I wanted to leave you with this idea that medicine for its entire existence has been the detection and treatment for neurodevelopmental disorders at birth, psychiatric disorders in adolescence. And when we get older, at clinical onset, the future is very different. Everything can be seen at birth using a brain and a disease model, and it gives us unprecedented opportunities to intercede well before a susceptibility becomes a clinically diagnosable disease. So the typical thing you're told is genes are the primary determinant of health, brain health. The answer is no, it is G plus E plus N. That is genes, caught the trigger, but actually what we're finding out is the environment and nutrition and a number of different kinds of environmental issues are actually the drivers of the onset of clinical disease. And so these are addressable issues if you can get the diagnostic early, and we can have, shift this to a curve that might not as steep, but much less steeper depending on the disease and the individual, and what is really going on. The business development forecast, I told you we have diagnostics and therapeutics in our crosshairs. We have 5 verticals. Each of them in themselves is a very large, addressing a very large need clinically, all of which are not being addressed effectively. The stakeholders are the federal government. I will show you that some of them have actually funded us now. We are looking, private philanthropy is what began our company and the ability to make these models. And we are talking to private family office now and private VCs and also pharma. This is a grant that we got, a phase 2 award now. It's for 2 years, 1.6 million from NIDA for our work on addiction. We're very grateful to the NIH for moving entrepreneurial science forward. We have biomarkers now that are patented and licensable. for various applications, and I've shown this before. And our business model shown in green is where we think we will reach market revenue. We think, for example, we can get market revenue even now using our well-calibrated disease models for R&D work. But then for everything else that requires FDA approval for medical device or IND kind of stuff, the market entry will be a bit more delayed. But once that is in place for all the different diseases, we think both using licensing and direct sales is when the revenues will start in approximately 3 years from now, once all of these things are accomplished and we reach the metrics needed to get FDA approval and bring it to the clinics. The
Rene?
capitalization, sorry.
Rene,
Yeah.
would you mind, because I think it's seriously significant in terms of the NIH doesn't vote with their checkbook unless they think that it warrants their checkbook. And, clearly they write checks. Can you talk about a little bit what caused them to vote with their checkbook in a significant way with you?
Yes, absolutely. So one of the crises that we are facing in the nation is the opioid crisis. And what is becoming apparent is with time that there are genetic susceptibilities underlying our response to the opioids. Some of us, and so our market was about the use of opioids in pain treatment and how post-surgical pain treatment has led to some people becoming addicted to the oxycodone, for example. And then when the clinical oxycodone is cut off from the physician, they have entered into the wild, wild world of illegal drugs. And ultimately, many of them have succumbed to it because those drugs are laced with fentanyl and all sorts of things. So our first product that we're trying to bring to the market is a diagnostic platform that can tell the physician before he takes a patient on into post-surgical treatment, pain treatment, whether or not they will have an adverse reaction to the drugs that are used for him. And so that ability to do that using drugs and the data we provided the NIH convinced them that we had entered the domain of a real commercial viable product that can be brought to the market. And that's what in phase 2 is being scaled and validated so that we can provide this platform, if you may, to address one of those things. But it also has use in psychiatric medicine where they're using similar drugs, and also in recovery programs where we can predict who will and won't recover as easily. So there's a lot of predictive medicine applications for just that one aspect that NIH has funded us for. I think you can just say rinse and repeat for all the other diseases. Very similar things will play out.
We have a question from the audience. They're asking if you could elaborate a little more on what you mean by brain in a dish.
Yeah, so the human brain develops the human body develops from 3 germline cells in utero. And the brain develops primarily from what is called the neuroectoderm. What the brain in the dish is a proprietary strategy by which we are able to manipulate the milieu of growth factors to allow and guide stem cells along a pathway that leads to the formation of various structures of the brain, to the point where over 12 weeks, it becomes a fully legitimately mimic of an early replica of the human brain. It's not complete and it's not quite the brain, and that's why we are called organoids, but it's sufficiently complete that when it's taken from a patient with Alzheimer's, it actually recapitulates the exact same program that led to the disease in the first place. And how do we know that? We measure the biomarkers that are dysregulated in our model, and we look at the literature of published genes that are dysregulated in postmortem human brains, and over 300 of them are identical to the ones we said are the drivers of Alzheimer's. So that's in a gist what we mean by a brain in a dish.
Thank you so much.
Yeah. Okay, so any other questions? So in terms of capitalization, we have, as I mentioned, a phase 2 SBIR, fast track, to address the opioid crisis. We have gotten money for COVID-19 from the CARES Act to take on COVID and how it infects the human brain. It does. We have planned strategies to get capitalization through various federal agencies, one of the stakeholders for either NIH or DoD. In fact, we are now approaching PTSD to address the needs of our veterans with PTSD. As I said, more than 22 of them commit suicide a day and have been doing that for at least the last 5 years. It has not been addressed. And so we're bringing our cutting edge technology to bear on a absolutely huge, huge problem that the DoD needs help with. In terms of the short-term financial forecast, academics is about 64 million a year in about 2 to 3 years. Corporate 80 million. Long-term is 10 to 50 billion. And we are currently have a stage of technology. We have patent applications, 9 of them, that cover many brain disorders. Key performance indicators are proprietary platform metrics, highly reliable, robust, and reproducible. Data is corroborated by clinical studies. Our current investment opportunities for those who want to invest with us are to be deployed for R&D and market entry, early diagnostic platforms, novel target validation, and also pediatric formulations as a new way to start to address at the pediatric level things that might be useful to both delay or prevent the trajectories that lead to disease. I said we have 9 patents applications and exclusively held by us, and they cover a wide variety of diseases. And what I would like to do is not go into all the details that a truly interested investor might be interested in expectations, addressing competitors, limitations, and also emerging medicine and its value, all of which I'd be happy to discuss one-on-one with the interested investors. And these are our distinguished clinician collaborators, advisors, and beta clients. The MD Anderson Cancer Center and Brain Cancer, Clay Marsh on the opioid countermeasures, Dr. Peter Cruz, who's a member of the Royal Society of Medicine, now lives in South Africa. And this is our team. The 2 of us are scientists, Susan McKay and myself. The other 2 are business colleagues. One's the dean at the Case Western Reserve Business School. And Johnny is right now in Canada. And whoops, I think I jumped the gun. What I would like to leave you with, and I wanted to just say something, is that there is so much more to what we're doing that meets the eye. And I wanted to leave you with a musical choreography of a symphony of neurons in what we have produced. And this is just my artistic side coming out. If we allow me some freedom to take science into another domain, what you're looking at are the individual neurons in the organoid set to music as we are scanning in and out of the organoid. And you can see very well each of these networked systems. We are now looking into the secrets of what goes wrong within each one of them. And to us, disease reflects a cacophony of the various neurons when they are not functioning in symphony. And so I'm bringing in a musical metaphor of our job is to correct somebody in the symphony whose violin is not in tune, so to speak. And by tuning them correctly, we bring back the symphony of what it means to exist, what it means to live life healthily, and what it means to give everybody the opportunity to reach their full potential.
So Rene, that's awesome. We couldn't hear the music. Was there sound?
So that was my fault. I think you need, when I did the share, yeah, that was, sorry.
It's okay. Take your time. We got plenty of time.
Yeah, it's.
I love the metaphor, by the way.
Yeah, it reflects something much bigger than any one of us is.
Take your time.
Yeah, there is a share sound feature that I didn't click on. I just did. And I will, for an encore request, play it again. Let me know if you can hear the music.
There we go. We got it now.
I choose classical music because it gives you a sense of the profoundness of what we are looking at today and how it'll change our future. It'll truly affect something that we've all struggled with for thousands of years. And the brain has been a sort of a black box. We didn't have access to the tissue, so we could never do anything meaningful in addressing brain disorders, but that's changing. Okay, I'll stop there. And I just wanted to say we have a full well-developed business team and a legal team represented in Chicago and Boston, both at the business level and intellectual property development. And also our accounting firm is Blue & Co. in Columbus, Ohio. So that's my contact information. I'd be more than happy to address either scientific or business questions from the audience, or you, Arthur, or Wendy.
Hi, Rene. We've gotten a few from the audience, if I could. One audience member is asking, when you speak about Alzheimer's biomarkers, you mentioned you identified 2,000. How many total are there? And so what is the percent of that 2,000?
Yeah, so it's a very astute question. When you look at the postmortem brains, you get a subset of biomarkers that is a snapshot at the end of the disease. And so there are about 300 of those that we capture in the 2000. The remaining genes are the ones that gave rise to those 2000 that you cannot capture in a postmortem brain analysis. And that's the beauty of our system. It's live living, and we can follow all the early markers that are, it's a domino effect. Eventually they get to the point where the brain starts to fail, and cognitively those things affect what we describe as Alzheimer's. But what precedes that, which is what we call early biomarkers, are not knowable using postmortem brains. And that's what our model systems allow you to do. And also they also give you insight into where to intervene to prevent reaching the clinically diagnosable time frame in which you are considered a patient for Alzheimer's.
We have another question from Paul, and I may slaughter this, Rene, so if you need me to repeat it, I will. Does the Organoid from an AD patient have all neuronal and glial types? Does it produce amyloid and phosphorylated tau?
The answer is yes. The answer is yes. And more importantly, we are the only group out of all the people trying to make organoids that actually have the microglia. And this is very important in Alzheimer's because the microglia has been considered the failure of the microglia, dysfunction of the microglia is considered one of the drivers of Alzheimer's disease. We are the only ones who are capable of studying it. We are. And other models that's where our competitive edge is. We know how to make a completely intact organoid with microglia. And we have evidence for that at various levels.
We have another question asking if you could speak more about the relationship to immunity and inflammation that you've revealed through your research and analytics.
Yeah, that's a again, another insightful question. We see the innate immune system involvement in many, if not all, of the brain disorders we have seen. They include drug abuse or substance use disorder. They include definitely Alzheimer's, but we see that even in autism. And so it is very likely that this is an issue that needs to be addressed seriously when it comes to the contributors to brain disorders. There's a common factor in all of them. Whether you define that as inflammation or infection at this point, I don't know, but it's certainly infectious agents being contributors to brain disorders is to be considered seriously. And there's a lot of evidence, not just from us, from others for over a decade that those types of findings using postmortem brains and other strategies have been published by very, very smart scientists. So we share in that belief, and our model system provide us the evidence that that is in fact going to be the case.
Additional comment, just letting you know that the images and music were beautiful. I agree, that was lovely. I have a question myself, if you don't mind, and then I have another one from the audience. But I see that you say schizophrenia, but there's a lot of schizoid affective disorders as well. Is it eventually going to apply there? Because when I watch the commercials on TV at night about great medicine, it's going to cause death, dementia, I mean, it's horrible. And I know a lot of those diseases come on with puberty. So will it affect them, and will it be able to get to them early enough where they don't have to take all these horrible drugs?
Absolutely. This is the excitement we have, not just that we have made big breakthroughs, we can publish great papers. The reality is much of what we thought was a one-way street of death and destruction, as you described, and desperation and tremendous suffering was unnecessary. It was unnecessary because we didn't know what to do. From our drug discovery program, we are discovering something very profound. The proper nutrition plays such a big role in the development of the brain, and in particular, there are reasons to believe that are also contributing to severe mental illness. Schizophrenia being one of them, autism being the other one. So we are now diving deeper and deeper into how does the genes and the nutritional aspects of it connect, and what kind of advisory or prophylactic strategies can be used without intervening with any drug to change that trajectory. And the sooner we do it, the better off we'll be. We foresee, I honestly think we foresee a baby wellness program at birth where we will know everything we need to know using our type of technology. And then from there, we will, through various strategies, including real drug development, begin to address them early so that you don't have to deal with what my generation and yours and everybody else has had to deal with, because we really didn't know much about how these diseases came to be, and we didn't have the tools. The tool is changing everything. So I'm very, very happy for the next generation. They will not go through, just like polio, most of us don't have it today, but thank God somebody came up with a vaccine. Otherwise, the iron lung would be a cure today. It's very similar in what I say, metaphorically use aviation. They're going to make a quantum jump. And we want everybody who's interested in having a legacy in doing that to join us in whichever way you think you want to be part of a revolution in medicine.
We have another question from the audience, and thank you, by the way, for taking my personal question. The question is, what is your valuation and what technical risks are of the most concern going forward?
We would like to discuss the valuation privately with the investors because of where we are in the pre-market analysis, and we don't see any real challenges, to be honest, when it comes to the science and development of the product lines. I think the biggest challenge would be to take an older form of medicine and replace it with a newer form of medicine. And those are market and political challenges more than a real scientific challenge And I think the NIH is trying to do its best to bring in personalized medicine. So all the forces are aligned. I gave you 2 examples of investors doing the same thing earlier on, where they have invested large sums of money in Series A type of funding to make this happen. So that would be my best guess as to where the challenges are really political and status quo. We are about changing the status quo. So it's challenging, but we think there's a good end goal, so we're going to make it happen.
Fantastic. We have another question asking for the names of collaborating academic institutions, if you're able to share.
I shared with you, we have institutions with MD Anderson Cancer Center with one of our advisors on brain cancer, Pudavali. The West Virginia Medical Center, he's the CEO, Clay Marsh. We have scientific referees who I did not actually feature here, but we have them at Cleveland Clinic. We have them at Yale. We have them at the University of Pennsylvania. Marina Pisiota is at Yale. Dr. Karis Eng is at Cleveland Clinic. And then we have Amita Segalo, the HHMI investigator at UPenn, all of whom are scientific referees for our work, and would be very happy to provide a scientific evaluation of what our work means and why, why we are one of the leaders in the field. Plus, we provide data transparency on everything we do once we have serious investors under an NDA. So you shouldn't invest blindly in anything without having it kick the tires and take it for a drive. You'll see this is the real deal.
We have another question asking what alternative technologies are a threat to your approach?
They're not a threat. There are ways in which you can get some of this information from postmortem data. There's lots of work going on single-cell RNA sequencing and RNA sequencing, but I told you it has limited value because it is a snapshot at the end of the disease. They do not have a way to recapitulate the earlier events. That's the big difference that we see. The other threat, and they're not really a threat, there are many people making organoids and they make parts of the brain. But when you look at all of the data published, we found out that they're seriously flawed. I'm very sorry to say that. They lack many of the biomarkers. They do not have the critical cells like the microglia, which are the innate immune system. I answered a question before how innate immunity is at a contributor, how majorly it is in different diseases varies. But a contributor to disease, when you don't have all the right types of cells in all the parts of the brain, you certainly will not get actionable predictive medicine outcomes that'll cut the mustard with, say, for example, the FDA. And it would be dangerous to use that kind of information to derive drugs to go into clinical trials. So we're not about just academic work. We're about making certain what we say has the ability to change the lives of patients in a predictable way.
We have a follow-up question asking what is the % accuracy of your predictive analytics?
We can only measure it as a function of clinical sciences because we are trying to model a disease. The Alzheimer's gives you a pretty good sense that it has a very high accuracy. The internal replication and reproducibility data that I showed you on those plots where we straight lines shows that we can measure all 13 or 14,000 genes expressed in the organoids with a R-squared coefficient of 0.9495. So it's very, very good. And we can make these organoids reproducibly time and time and time again. We've done it for a large number of brain disorders. So we realize that the QC is the key to getting good information. Only then can you use AI or machine learning to infer what you need to know. As somebody who's trained in the physical sciences, I come from quantum chemistry, I know that models are model and modeling is modeling and AI is AI, but we have an old saying, garbage in, garbage out. So we make sure that what goes into those machine learning techniques are highly curated, highly predictable data sets so that we don't have to deal with unpredictability on the other end.
Sorry, there's trying to get myself unmuted. So if anybody else has any questions, please post them. Otherwise, Arthur and I are going to ask questions. And I have a bunch, but Arthur, did you have some?
Just one for now. I have a bunch, but Rene, are you able to, without revealing things that aren't appropriate to reveal, except under NDA, talk about the amount in this round that you'd like to raise?
Yeah, I am trying to raise about $10 million, and it's based on the fact that we have 5 verticals, and we would like to have all of them reach, rather than doing that serially, we want to do them in parallel so they reach the FDA metrics we need, at which point you open the door for everything from drug discovery to diagnostics and everything else that can be brought to the market. So I would like to make that all happen for all of them as soon as possible. So I think a $10 million initial investment will allow us to scale and get to the end points, because the technology is absolutely perfected in our hands. I wouldn't be saying this as a scientist. I know that's very difficult to make the claim using a biological system. But we put a lot of time. It's taken 10 years to get here. So there was a lot of QC and all the ways in which things can go wrong were all figured out. And so what we are now able to do is produce highly reproducible data. So the 10 million is our initial estimates, and we are quite open to syndicated investments if that's how people want to do it.
Has the NIH decided that they're going to step up again, or is that not necessary?
The NIH is a federal agency. They are very interested in making things happen. They provide the phase 2 money to bring things to a point where investors are now able to carry the next steps to bringing it into the market for commercialization. They themselves are not the ones. There are phase 2B grants you can apply for, which is something we will do. If you go with DoD, there are phase 3 grants you can go with. I think some of them are, if you're going into clinical trials, they are as big as $10 million a piece for anything else. That is of interest to the veterans in the U.S. Army. And in fact, PTSD is something we are very aggressively pursuing. We already have a fair amount of information on that makes us confident that that is something we can address.
Wendy?
Hi there. So I had a couple more questions, and I'm happy to follow up with you after, but this is just fascinating. I think a lot of us have had family members that either, were in the services, had problems there, had Alzheimer's, had other kinds of issues. So are you able, I understand the concept of being able to take a baby's brain, be able to go in and say, we're going to predict you might have X, Y, or Z. I'm a little less sure, and it could be because I am not a doctor, but I'm a little less sure how you take somebody who may already have this in a more advanced state and bring them back. Is that possible? Is that where it's headed?
I can't answer that question without knowing whether the implementation of what we find out actually works in an adult late stage Alzheimer's, for example. It's kind of like saying, after you get polio, will the polio vaccine work, right? So our goal, and even it including myself, I'm over 60. So there's no certainty that I will be able to cure what I might get before I get it. But I think we are about my generation, the way I see it is, we do that nonetheless. We provide and change the future for everybody we can. And so depending upon the disease, depending on the state, depending upon what was the driver or the condition under which you ended up in a clinically diagnosable form of, say, Alzheimer's or autism, they may in fact be unbelievably simple ways to address them. And one of the things I'm going to tell you is we are looking into autism, right? We see, and I'll openly disclose to you, we see that there are a vitamin B6 deficiency. And you kind of go, well, it should be simple. Go to GNC, get vitamin B6 and be done. It doesn't work that way. What we see is that there are enzymatic pathways that convert B6 into the biologically accessible forms that help you develop your brain correctly. And those enzymes, they call PLP enzymes, the genes for them are dysregulated. So guess what? All the B6 in the world will not help you. But if you have the biological form of this given to you at an early stage in your life or even later in life, it can have profound effects for pennies. So this is why I think it's important to allow the science to get to the point where all the components that give rise to each disease, and it might vary in subtypes, even within autism, for example, within Alzheimer's, that information provides each individual with their nutritionists, their family practitioners, with their psychiatrists to change their basic needs metabolically, and then add adjunctively the known therapies that work, cognitive behavioral therapy maybe. It's kind of like when you go and get your knee replaced, you still need physical therapy to get everything back in order. But if you didn't have the knee replaced, no amount of physical therapy would change the issue that is underlying the loss of your meniscus or tears or whatever else. So we see we see what we do as the ones who are going to go and surgically fix things, so to speak. But all the other adjunctive techniques will also play into the recovery process for each of the diseases that are already functioning in med. So it's going to just add value and make things better earlier, sooner, and better. So I know you showed an interest in schizophrenia. Are you willing to say why?
Sure. And some people who are on here know me very well and know my children were born tox pos, and they as they've gotten older towards adolescence, they've developed some of these disorders. And because we don't have a genetic history, it has been challenging, tearful, joyful, and everything else you can imagine discovering, and then having to work our way through, because we're not doctors, medications and how to function. And I would have given just about anything to be able to have them tested at the hospital to know it was coming and been able to head it off, help them.
Well, we can talk after this. We can talk after this if you wish to. I work with families whose children are afflicted by autism a lot. And we are doing a lot of things together to understand and provide the kind of information we need and they need to make their children's lives better. So I'd be more than happy to talk to you to see and bring to bear some of this kind of information. If it's a simple vitamin, we don't hold that back from anybody. We tell you, look, this is something you got to do for your child, but we don't know what's going on in any individual family. But I do that. I work with them, and it helps develop our science to be a better science to address pediatric needs. So that's where we started, actually. If you recall, I said we started with autism.
Yeah. Well, I thank you very much. Arthur?
It was very kind of you, Rene. Thank you for that. Wendy's a dear friend, so I appreciate the offer. This has been fascinating as always. Like I said, we're going to make sure everybody gets in touch with you directly. And you did a really outstanding job of explaining a very arcane information in a way that even I could understand it. So I'm going to take you up on your offer and not fly over Ohio. I'll stop.
Yeah, we're not the flyover state. There is real good science happening here, and we are nested in a very good place. And I think we are excited to share this with the community and also with the country, and maybe even globally, that there's real hope. It's based on real science, and it will make a difference. I'm absolutely certain of that. And thank you so much, Arthur, for giving me the privilege of being on your investment forum today.
Yeah, it's my honor. Let's see. I think we have maybe one more Oh, somebody, yeah, I was just saying thank
Sure.
you. It was super, super nice. So, Wendy, thank you for joining in today as usual, and really appreciate your questions and input and helping us along here. Rene, again, thank you. Fascinating
Sure.
presentation. We'll make sure everybody's in touch. And remember, we're recording this, so if there's anybody in your community here from the attendees, you're welcome to share that. And also feel free to connect with Rene directly, and we'll make sure that he has all your information. Super interesting. Congratulations. Much
Thank you.
success. And then I'll be talking to you separately anyway, Rene.
Okay, yes, look forward to that. Both Arthur and Wendy, after this, we can connect and see what we can do.
Perfect. And as I always say, thank you all for joining in today and sharing with us the only thing you can't make more of, and that's your time. Till next time.
Yes, thank you. Bye-bye now.